Welcome to Chapter 15, talking about genes and proteins. And that means we're going to be talking about how a cell gets from the information that is held in the DNA. It will go through the intermediate of RNA to finally get to the protein, that flow of genetic information in cells. And I do qualify that as in cells because viruses do other crazy things that if you're interested in, we will talk about in a microbiology class. We are sticking with cellular biology, so we will talk about how DNA is interpreted through RNA to get to proteins. I have included several resources from the Khan Academy that line up very well with our Chapter 15. There's brief videos, there's articles to read, and there's short quizzes that let you check your understanding that are graded within the Khan Academy. Again, those don't count toward a grade per se in your class, but if you have reviewed these resources, you will be in very good shape for the types of questions that you will see on our next exam that will cover Chapter 15. So there's videos on transcription and translation, and there's some good articles as well about those processes. And looks like the title of this got cut off, but this is just showing and reminding us that the macromolecule of proteins are polymers of amino acids, that all proteins have those three levels of structure. The primary structure, that is the sequence of amino acids. The secondary structure, where we have hydrogen bonds holding regions of that polypeptide chain into coils called alpha helices or straight ribbons called beta sheets. The tertiary structure is an interaction between those localized regions of secondary structure. And the tertiary structure is held together by covalent bonds. Those would be disulfide bonds between cysteine amino acids, hydrogen bonds, van der Waals forces, and ionic bonds. And that 3D structure that results from the tertiary level of structure is the final shape of that polypeptide. Many proteins only go through those three levels. Some proteins are made of more than one tertiary structure that then have to interact. And in that case, we have the quaternary structure. And that's two or more tertiary structures interacting, and they will be held together by the same kinds of bonds that you see in a tertiary structure. Our genes are on our chromosomes, and our chromosomes are made of DNA and those associated proteins that we saw in the last lecture. And we see this is the interleukin-2 protein. Scoop me out of the way for a bit. Interleukin is an immune modulator, a chemical secreted by immune cells that helps to orchestrate the overall immune response. And C is alpha-2U globulin, so this only has one tertiary structure. Hemoglobin has 4 independent tertiary structures that have to come together to build the final molecule. So our central dogma of molecular biology, how we currently view the way things work, is the information flow. That we have the DNA is the original blueprint, the instruction manual. We have to have the intermediate of the RNA that the cell can then interpret to make proteins. So DNA is that blueprint. If we don't have RNA, we don't have the instructions that the ribosomes need to make those proteins. We saw in the previous chapter that DNA replication required DNA polymerase I and DNA polymerase III to make an exact copy, and that process is called replication. In this chapter, we're going to see how does the cell interpret DNA to get a copy of that genetic information into the molecule of RNA, and that is done by the enzyme RNA polymerase. So we're an enzyme building the polymer of RNA. That RNA polymerase will bind to DNA at a site called the promoter. And by site, I mean a precise sequence of nucleotides. And the order of those nucleotides, they are chemical, so they will have a particular shape. RNA polymerase will recognize the shape of that promoter series of nucleotides, and that will produce an RNA strand, that has a sequence that is complementary to the DNA. And we will see images of that to see what I mean by complementary. And that process of RNA polymerase reading that DNA to create RNA is called transcription. We do not turn DNA into RNA. The DNA is unchanged by this process. We use the information of DNA to create an entirely new molecule, that is RNA. And then finally, in this central dogma, ribosomes, those cellular structures made of ribosomal RNA and protein, form a little sandwich around messenger RNA, and that will direct the cell which amino acids to deliver, create peptide bonds between those amino acids, and make proteins. That process of reading RNA to create a protein is called translation. And again, we do not convert RNA into proteins. We use that genetic information held in the molecule of RNA to direct the synthesis of protein. Copy of DNA is replication. Reading DNA to make RNA is transcription. And reading the RNA to make proteins is translation. Here is a little better view of what a promoter is. This series of rectangles is representing DNA. This region here, these 4 boxes, is the promoter sequence. The giant yellow blob is the RNA polymerase. Other labels, we have the +1 transcription start site, and we have -10 and -35 regions. This +1 says this nucleotide right here is going to be the first nucleotide in the DNA that will direct, that will be the first nucleotide of the RNA. There is still DNA here in the promoter region, and we've got -10 means 10 nucleotides, let's get my thumb in the picture, 10 nucleotides back from the start of transcription. The -35 region means 35 nucleotides back from the start of transcription. The -10 region is inexplicably called the Tata box. It's like a Tata at, T-A-T-A-A-T. That is the region that, starting here, is 10 nucleotides that way from the start site. And the TT-GACG, a little bit of variability here, but this region is conserved. Always the same, whether it's in eukaryotes or prokaryotes, which eukaryote, which prokaryote. That sequence is the signal for this big RNA polymerase to bind over this whole region, and once it hits here is where it will start the transcription. That's our RNA polymerase again, the big sort of tannish yellow box. Here we're actually showing the 2 strands of DNA. I'm sitting up in bed recording. It's a little lumpy. 2 strands of DNA. They are labeled by their 5' and 3' ends. However, unlike replication, the RNA polymerase doesn't have to have that free 3' hydroxyl. RNA polymerase has its own enzymatic capability to break the hydrogen bonds between the two DNA strands, and once that has happened, catalyzes the addition of the ribonucleotides to build RNA. We could see the DNA sequence, even if it wasn't labeled as DNA. We would know that it is DNA because thymine nucleotides are present. And this pink ribbon is the RNA because it has uracils. So anywhere in the DNA where there is an A, the RNA polymerase will add a U as the complementary base. So A's pair with U's. If there's a G in the DNA, there's still a C. If there's a T in the DNA, T still pairs with A and C's with G's. So CG pairs are still the same. A T in the DNA will direct the position of an A in the RNA, but an A in the DNA says the RNA has to have a U in that position. And as the RNA polymerase moves along the DNA, it opens it up, synthesizes the nucleotides of the RNA, and then zips the DNA back together. We have two labels on the DNA strands. We have the template strand and the non-template strand. So this polymerase is only reading the bottom strand. It is not reading the top strand. The template strand does have a free 3' end, and that's because we build the molecule of RNA starting from the 5' end and build it to the 3' end. And there are mistakes on this image, which is once again proves why one should proofread. This should be a U because we're on RNA. So this should be a U. This should be a U because this is the RNA molecule. Can be relieved that I didn't create this slide, but I did not proofread it to catch that. Can't have an ATG in RNA. That has been AUG, an AUC, a UGU. All the rest are okay. So the template strand on the left side has the 3' end because the RNA is going to start being built from the 5' end. And the RNA polymerase will continue until it reaches a sequence where it causes the RNA to do a bend, and that pushes the RNA polymerase off of the DNA. This is a generalized promoter. Here is the +1. We've got the, this should be the -10, TATAA, that's the -35. We don't just bind the RNA polymerase directly. There are transcription factors, that's the TF. So this is transcription factor 2, that's a Roman numeral 2, D. And then there's a transcription binding protein that triggers other transcription factors to bind, and then we finally get the RNA polymerase. All of that has to come together to initiate the RNA polymerase to be able to actually get to that +1 site. So in our eukaryotes, in addition to having linear chromosomes, and all DNA is going to have those promoter sequences, eukaryotic DNA has both coding and non-coding regions. Coding means it's going to end up in the final RNA that a ribosome will read and make protein. Non-coding means it's going to be removed and will not appear in that final RNA. So those coding sequences are called exons because once transcription is complete, the RNA gets processed and the exons get to exit the nucleus so that they can get to the ribosomes that are either free in the cytoplasm or are on the rough endoplasmic reticulum. Introns are the non-coding region because they get removed from the initial RNA transcript and they stay in the nucleus. So exons exit the nucleus so that ribosomes can read them. Introns stay in the nucleus and they get digested and their nucleotides get recycled. The DNA contains the promoter. It contains an entire sequence that contains both exons and introns. The whole thing will be transcribed to make a long RNA that represents all of the coding and non-coding regions. That's called the pre-mRNA. mRNA stands for messenger RNA, the RNA that's going to be the message that ribosomes get to read. We get what's called a 5' cap, a single guanosine nucleotide that gets stuck on the 5' end to protect it. We get a 3' poly A tail because there are multiple adenine ribonucleotides attached to it. And again, that protects it. UTR stands for untranslated region. These are nucleotides, but they are not going to be read by the ribosome to get translated. Once we have all of that, there are enzymes that are going to do splicing. To splicing means to cut and then reseal the leftovers. So the RNA splicing will cut out the introns and seal up those exons in order so that our final messenger RNA has only exon nucleotides in addition to its cap and tail. And then the ribosome will start reading at the first nucleotide that is part of an exon and will stop when it gets to this untranslated area. Prokaryotic DNA does not have introns. There's only coding sequences. Eukaryotes have introns and exons. Initially, there was the thought that this was junk DNA, but we have so much of it, and it is energetically extremely expensive to synthesize it, that there are other hypotheses as to its roles, and we'll get into that when we get into control of gene expression. So there is another cartoon showing that our initial primary RNA transcript contains both exons and introns. We get our little cap and our tail, and the introns are removed and stay in the nucleus. The exons seal together and they go out. They exit the nucleus. The way that splicing happens is very precise. There are small ribonucleotide binding proteins that cause this loop of the intron and cut it at the ends of the intron so that the enzymatic activity of them will seal the exons together and the introns leave as this looped structure. That sets us up for the second half. The transcription is the first part of Chapter 15. So Chapter 15 will be broken up into two relatively short lectures because there's a lot of information in it. But I do want to end this part by talking about codons and reading frames, because that will set us up for understanding how the ribosome is going to sandwich around that messenger RNA and be able to interpret that genetic material as instructions for building a protein. So a codon is a sequence of 3 RNA nucleotides in a row, and they encode or they specify a single amino acid. And each codon will only encode one amino acid. So if you have an AUG, there's only one amino acid that the cell will recognize should be delivered to that AUG. If you have a CCC, there's only one amino acid that can be delivered as being instructed by that CCC. However, one amino acid could have more than one codon, and we'll see that figure in a moment. The reading frame is how we expect those codons to be read, and the start signal is always AUG. That will always specify for the cell to bring methionine in eukaryotes and a chemical called formylmethionine in prokaryotes. It's still the same amino acid, it just has an additional functional group. So AUG is a signal that tells the cell, the ribosome, start here. And that reading frame is always 3 in a row. 3 in a row, 3 in a row. So if we have an intended sequence of an AUG, there's one codon, a CCU, a GCA, and a UAG. If we insert just one letter, this little red G, first codon is okay, the second codon is not, the third codon is not, the fourth codon is not, and we have a single letter left over, that's no good. It's not a codon. If we have a single deletion of this first C, again, our AUG is correct. We should have a CCU, but we have a CUG. We should have a GCA, we have a CAU. We should have a UAG, and we have an AG, which is, again, not an entire codon. So that's what's meant by the reading frame. We frame the codons in these sets of 3 that have to be that 3 and in that order. So a single insertion or a single deletion throws off the reading frame at that codon and every single codon after that. We could have a substitution where we change a letter. As long as we don't change the number of letters, that may have an extreme effect or no effect at all. So this is our codon chart. Please do not memorize this. I have exactly 4 codons memorized. I know AUG is methionine or start, and I know that these 3 tell the ribosome to stop. Everything else, I gotta look it up. So there are 64 codons. However, there are only 20 amino acids. So 3 of them aren't amino acids at all in this codon, so that leaves us 61 codons for 20 amino acids. There's only one that's going to give us methionine, but when we look at this, let's look at this LEU. I will give you a codon chart that actually has the whole name spelled out, but this stands for leucine. And if you have UUA, UUG, or any of these, all 6 of those letter combinations still give you leucine. See, what else do we have? One that's got a whole bunch for. Proline has 4. I think there's another one. Arginine has 6. Those 4 and these 2. What you'll notice is when there's a variety, let's look at serine. You see in the first 2 positions, and anything in the 3rd position, you still get serine. An A and a C in the 1st position, and any of the 4 options in the 3rd position, you still get threonine. This effect is called wobble. Our genetic code wobbles because the critical thing that results from our genetics is this amino acid sequence, the proteins that actually go out and do the functions in our cells. So making sure that we get the correct peptide amino acid sequence is critical. Having more than one way to get to the same amino acid means our code wobbles. It means we are protected. If there was a mutation in the third position of the codon, no matter what that mutation is, we still get the correct amino acid. That means that if there's an amino acid that's really critical in the folding and the overall positioning of amino acids and the final structure, we have to have that one. It's probably going to have a lot of ways, a lot of different codons to make sure that even if we have mutations, we're going to get the correct amino acid. And the amino acids that have the most codons tend to be the amino acids that show up most often in the active site of enzymes. And that active site is the position that carries out reactions. So it's critical to get that correct. Next time when we actually look in how is a ribosome going to interpret this and build a protein, we will also look into, well, what happens when we don't get the expected nucleotides? How does that affect the function?